So also, I've been thinking about how to put this number in perspective. Our average is our estimated average population size or mean population size is 5.5 million bees. So I was thinking, how do I compare that to things that people are familiar with? So you've mentioned the honeybee comparison. I'll also point out that it's 4 times the human population of Manhattan. And if we calculate the biomass of this single, generation of emerging bees, it's 4,000 kilos. So it's 4 metric tons, which is equivalent to 4 adult giraffes. No, sorry, 2 adult giraffes. So imagine 2 adult giraffes grazing in your local cemetery. That's the Andrena regularis population right there.
From the Oregon State University Extension Service, this is Pollination, a podcast that tells the stories of researchers, land managers, and people like you who are making bold strides to improve the health of pollinators. I'm your host, Dr. Adoni Melathopoulos, Associate Professor of Pollinator Health in the Department of Horticulture. Many of you know that solitary bees don't live in massive bustling colonies like honeybees, bumblebees, or certain sweat bees. But just because they are a solitary bee that nests alone, it doesn't mean they can't draw a crowd, especially when thousands of them build their nests right next door to each other in giant aggregations. Joining us today to dive into these buzzing neighborhoods is Dr. Bryan Danforth, a professor of entomology at Cornell University. Now, Dr. Danforth is the lead author of The Solitary Bees, and some of you may remember that was a book we previously featured on episode 107. And he's back with us to discuss a fascinating new study published in the journal Apidology. The study was led by Steven Hodge and is titled Parasite Dynamics and Host Parasite Associations in a Large Aggregation of Andrena regularis. Now, not only does this research reveal the largest nesting aggregation ever recorded for any bee species, but also sheds light, most importantly, on the complex population and parasite dynamics at play in these nesting aggregation. And there's also a twist. This massive nesting site was found in a cemetery, raising the wonderfully ghoulish possibility that our graveyards may double as crucial conservation sanctuaries. So without further ado, let's head down the ground nesting bee rabbit hole this week on Pollination. Well, welcome back to the podcast, Dr. Danforth.
Thanks very much, Anthony. It's great to be here.
Well, I was really delighted when you reached out to me. You have a new paper that you've published, and it's quite amazing. And it's in my mind as, I walk around, the campus here. It's springtime, and there are ground nesting Andrena that are popping out of the ground, sometimes in large numbers. But you, in this paper, you and your colleagues describe perhaps one of the oldest, but certainly the largest recorded nesting bed. Can you describe the primary bee species? I know there's multiple bees that were nesting in this site, but what was the primary species and what do we know about its biology?
The main bee nesting at this site is Andrena regularis, although there are Calides inequalis nesting in the same cemetery, and there's other Andrena nesting there. But regularis is the most abundant in the area of the cemetery we studied. And this is a pretty large Andrena. It's in the subgenus Melandrena, so it's a big black and yellow bee, beautiful, fluffy bee. This was a bee that was studied in Illinois in the 1970s by Schrader and LaBerge. So we already knew a little bit about its nesting biology, its foraging behavior, brood cell structure, that kind of thing. It was also studied in the 1990s by Malinsky and Osgood in Washington State and in Maine, exploring what its role is as a crop pollinator. So we know about its basic biology. We also know it's a very important apple pollinator. It also visits blueberries. So in Maine, it's an important blueberry pollinator. And in our work on bees of New York and our decade-long survey of the wild bees of apple orchards over the years, Andrena regularis ranked number 3 in abundance of the 120 wild bee species that we recovered in our surveys. So this is a whoppingly important bee for apple pollination. Oh, and also point out that it's a very effective pollinator. So there were wonderful studies by Mia Park looking at the per visit pollinator effectiveness of Andrena regularis versus bumblebees and honeybees and other bees. And regularis came out on top. It's something like 4 times more effective than honeybees in depositing pollen on the stigma of an open apple flower. So these are really good pollinators.
I remember Dr. Park had a nice extension publication that came out of this. It was at the time I was just hired and I saw it and I thought it was like one of the best native bee crop pollination guides I'd ever seen.
Oh, thanks. Yeah, she did a great job. She had she had extension funding to do that, and it's still being used. It's available online. Yeah.
Well, we'll link it in the show notes for people to take a look. It's really wonderful.
Thanks.
But I guess it's worth mentioning at this point the site. For most people, the site is not what they have in mind when they think of prime bee habitat. Tell us about this site.
So, yes, we did this work in the East Lawn Cemetery, which is just off the edge of the Cornell campus. Literally, I can walk there in 15 minutes from my office. So it's not like we had to go very far to do this study. And actually, the site was originally discovered by Rachel Fordyce. Shout out to Rachel Fordyce, a former technician in my lab, who parked her car at the East Lawn, East Hill Plaza, a little shopping center just beyond the cemetery. And she would walk through the cemetery every morning. And one spring morning, she brought in a jar full of bees, and it was like, these are all over the cemetery. What are these? That's how we got started on this project. So really, just a random and very astute observation by Rachel. So in a sense, that I'll describe the cemetery to you. It's a typical, cemetery. It's mowed regularly. It has low pesticide use, which we think is actually, a good benefit to the bee community. Pretty high floral diversity, lots of flowering trees. But what makes it special is that the soil texture at this site is sandy loam. And sandy loam is this kind of magical soil that we're finding lots and lots of bees like to nest in. Um, and it turns out, and this is my hypothesis, I have no proof of this, but we're going to start exploring it, is that when these old historical cemeteries were built, you had to dig a hole in the ground by hand. You had to dig it with a shovel. There was no backhoe back then. And so sandy loam is actually the soil that makes that kind of digging very easy. So I don't think it's random. I think that these cemeteries were sited in places with well-drained sandy loam soil, and that's just the same kind of soil that many ground nesting bees like. Of course, some like more sandy soil, some like more clay soil, but there are a lot of them that like sandy loam. So now my new project is to explore all the cemeteries in upstate New York that were built prior to the backhoe. So that's 1950. So we're going to look at all the cemeteries that were built prior to 1950 and see if they have, if they're often sited on this kind of sandy loam, and if they have bees.
I did joke with my wife that, I want a sandy loam historic cemetery to be buried in so that when the Adrena flies, she can think of me. But anyways, it's
I
like,
do. I did, just aside that you might find interesting. I did purchase 2 plots at the cemetery, so my wife and I will be there amongst the bees someday.
but it is an interesting idea. And I love the whole, people needing to dig just like the bees and, a relative ease, and that this might be, I think at the end of the paper, you describe it as maybe these are a really overlooked area. We think about conservation in certain ways, but, these cemeteries might be actually of high value.
Yeah. Well, I did dig into the literature a little bit, and there are some studies of cemetery biodiversity, particularly in Europe. So when you have, big cities like Berlin, and there's old cemeteries scattered throughout the city, these cemeteries often have rare plants and animals. So there is quite a literature on this,
Wow.
and not on bees so much, but more on sort of birds and flowering plants and things like that, that you can find these rare, they're preserves of biodiversity. And one of the great quotes that I found, I just love this, is someone described cemeteries as sentinels of biodiversity where the dead protect the living. It doesn't
That
get
is
any better than that.
great. Oh my God, so poetic. I guess the other thing to mention about this site is there are records of this bee, almost 100 years ago at this site. This locale, this bed might be quite old.
Yes, yes. Thanks to GBIF, we found a record of Andrena regularis collected at that site. And it was a farm before that. So before it became a cemetery in 1878, it was a farm. So there's a record of Andrena regularis. I think it was early 1900s, early like 1920 or something like that. I mean, Cornell has an insect collection that goes back to 1865. So likely the bee was nesting there, but we do know that it was present. They were flying in that area at that time.
Now, this study relies on emergence traps, and I remember talking to a colleague about emergence traps and putting them out in the landscape to try and like estimate, ground nesting bees and had very little to show for their effort. They were very discouraged by emergence traps. Now, your group has used it in a new way and I think to great effect. Can you explain how these traps were used in this study and, how you were able to, how it's a pivotal part of making these estimations of, the populations, but also the parasitic bees?
Sure. So emergence traps are little, imagine a little tent covering an area less than a square meter, and you can buy them from Bug Dorm. Just plug for the Bug Dorm company. And at the top of the tent, there's a collecting jar, which we put alcohol in. So anything emerging out of the ground, any bees or beetles or flies, fly up into the collecting jar. They're captured in ethanol. And we put out 10 of these traps in kind of semi-randomly in the area that we knew the nest site was located. And then the traps were checked daily by Steve Hogue, the first author on the paper. Steve's an undergrad at Cornell, was an undergrad at Cornell. He's heading to med school now at Rochester. But Steve did a great job on this project. In the spring of 23, he got the traps out early. He checked them every day for 40 days and really documented kind of the phenological emergence of the host and all the parasites that go along with it. So we used it in a slightly different way. There are I don't want to claim like to be the first person that's ever thought of this. There are other studies where people have used emergence traps to examine population dynamics, population size. In other ground nesting bees, there's a nice study in Brazil on Apicarus. So it's we're not the first, but we did think it was a good way to estimate the population size because it's really hard to estimate population size in ground nesting bees. You can count nests, but that's just telling you the number of active females. You don't know how many males there are. So this was the idea. Can we use these traps to do this? And luckily we knew the limits of the aggregation. It was pretty defined. So we could kind of throw down our 10 traps and in the following spring and then just, check on them every day.
Now, I think one of the most fascinating parts about this paper is not simply the bees, but the emergent patterns of parasites and predators of these bees. Tell us a little bit about what you found. It was really intriguing to me.
So yeah, one of the things we thought was interesting is that the emergence, you capture a very fine-grained picture of the emergence. So day-to-day variation in the number of males, females, hosts, parasites, flies, beetles, everybody coming out of the ground. The Andrena come out in a very big burst at the very first day of 70-degree temperatures. So you get males coming out slightly before females, but not much, a day or two and then the female peak. And so they're almost perfectly overlapping. So the Andrena comes out bang on this, first 70-degree day. And then there are a few short periods of emergence after that when it gets warm again. But the Nomada do this interesting thing where they kind of come out slowly. Like they come out a little bit on the first, warm day, a little bit on the next, a little bit on the next. So the Nomada seem to be spreading out their emergence. And we kind of thought about that, and I think my best explanation is that Nomada needs the host to be actively provisioning their brood cells for the Nomada to parasitize those open brood cells. So it kind of makes sense that the Nomada shouldn't be in a rush. They should be willing to let the host get to work. We don't know how long it takes from female emergence of Andrena to the building of her nest to the provisioning of her first brood cell, but it must be at least a week to 10 days. So that the Nomada, delayed emergence in Nomada kind of makes sense. And of course, if you dig into the literature, you often find that you're not the first one to figure this out. So we did find one paper on another Andrena and another Nomada that suggested the same thing. So it may be a general pattern. Parasites might want to emerge a little bit after their host to kind of give the host a chance to get busy nesting.
So in the other case that you were talking about, it was this kind of delay start. Or is this really the extent of our knowledge on the phenology of, cuckoo bees on their hosts? There's,
That's
literally
it.
a few studies. Geez, okay.
That's all I know. That's all I could find. I mean,
That's
Jerry
incredible.
Rosen, who studied brood parasitic bees for decades, and documented very interesting brood parasitic behavior, didn't ever really look at phenology. His studies were kind of done, opportunistically when an aggregation in a parasite was discovered, but his studies were done all over the world, and so you can't go back the next year easily and do an in-depth study.
I guess the other thing is that there, in addition to cuckoo bees, maybe our listeners don't know this, but solitary bees are hosts to, a number of other non-bee parasites and predators. You had some discoveries there as well.
Yes, we found a meloid beetle that's likely a brood parasite of Andrena regularis. We also found a conopid fly, which had been discovered by the folks who studied this bee in the 1970s. So we think that's a pretty likely association. Conopid flies parasitize adult bees, but then I think the adults go into the ground and then the conopid comes out. So yeah, but those were very low parasitism rates. What we do know about brood parasites of bees is that the bee brood parasites are the most impactful. Bob Minkley did a very nice literature review of mortality and ground nesting and stem nesting solitary bees, and he found that if you look across hundreds of studies, the bees are the most impactful. Like they can have parasitism rates of 10%, even higher. In our case, I think the Nomada was at a pretty low rate, like 1 or 2%, I think it was. But the beetle and the fly are even lower than that. They're a fraction of a percent. So again, the Nomada is the major source of mortality, but it's not a huge source of mortality.
I was also intrigued by the part there's an area of the paper, it's brought up in the introduction and the expectations around sex ratios, that sex ratios tell you something about a species and maybe its environment. You discovered the investment in females was larger than males, not the sex ratio, but if you just weighed them and kind of looked at the overall investment, that it was larger. Tell us, lay for our listeners out, why is this a surprise and what this might imply?
So I'm going to do a little bit of hand waving here,
Okay.
just FYI. But yes, so as you know, Hymenoptera are haplodiploid. So females can choose on a daily basis the sex of their offspring. So they can fertilize the egg if they want to produce a female. They can choose not to fertilize the egg if they want to produce a haploid male. So all the way back to parasitoid wasps and sawflies, Hymenoptera can do this magical thing where females can bias their own sex ratio one way or the other. And we know from lots of nice studies on bees, particularly stem nesters, that things like floral resource availability can impact sex ratio decisions. So when females have lots of floral resources, they tend to produce more female offspring. When there's limited floral resources, they produce more male offspring. Now, why is that? Females are heavier than males in most bees. Therefore, they're more costly to produce. They take more pollen and nectar. They take more time. So when females are stretched for floral resources, they will take the easy route and they'll make a male. They'll make a cheap male. And so we think that there's potential for this being a kind of indicator of population health, that when populations are more female biased, they may be, doing well. They may have abundant floral resources. Females may be making decisions to invest in the more costly sex rather than the less costly sex. Now, the contrary would be that if populations are struggling and facing limited resources, they should shift to males. And there's some evidence of that. But that's what we wanted to explore in this case. So we did put combine the body weight data with the numerical sex ratio data, and we found that they deviate significantly from equal investment. They're producing more females than males. So we take that as a good sign for this population.
That there's ample nectar and pollen, so they're not making the cheap males. They're
Yeah.
opting for the more valuable females, and this is maybe an indication of health.
Yes,
Huh,
exactly.
fascinating. Okay, so the big well, I think the big news in this paper is that you estimate a population, maybe almost 8 million bees maybe in this cemetery. Uh, I was fascinated with one of the comparisons in the discussion that, to put it into perspective, that would be the same number of individuals as 270 honeybee colonies in this really compact area, smaller than a hectare. So I guess this raises the question, how does a massive native bee population change our understanding of the typical native bee densities in agricultural areas that, especially in spring blooming crops where you would imagine a bumblebee colony you're only having queens, you just can't get the density. This seems like a challenge to that.
Yeah, that was kind of one of the things that we got so excited about. So also, I've been thinking about how to put this number in perspective. Our average is our estimated average population size or mean population size is 5.5 million bees. So I was thinking, how do I compare that to things that people are familiar with? So you've mentioned the honeybee comparison. I'll also point out that it's 4 times the human population of Manhattan. And if we calculate the biomass of this single, generation of emerging bees, it's 4,000 kilos. So it's 4 metric tons, which is equivalent to 4 adult giraffes. No, sorry, 2 adult giraffes. So imagine 2 adult giraffes grazing in your local cemetery. That's the Andrena regularis population right there. So what do I take away from this? I think that it's sort of just a message that we sometimes, we're often underestimating the abundance, the value, the importance of ground nesting solitary bees when we think about bee pollination across the landscape. So, this is a population of bees that's servicing the Cornell apple orchard. I mean, they're the major pollinators at the Cornell apple orchard, which is 500 meters away. So let's hope that other apple orchards in upstate New York have similarly large Andrena regularis populations, and they probably do. But I think that's kind of my take home is that it's a bigger number than I would have guessed if you'd asked me like what's a typical aggregation of ground nesting Andrena look like. I also think it has an important conservation message. If we don't identify these sites and preserve them and treat them with care, then we can eliminate an enormous number of individuals and that have been there for generations. So I really take it as a sort of conservation message. Let's identify these nesting sites. Let's get people interested in them, and let's figure out ways of focusing attention on their conservation.
Well, and I think this is a good plug for those of you who don't know of the Ground Nesting Bees initiative out of Cornell, the iNaturalist project. I'm so excited about that project, and we've had Jordan on in the past. It allows us to start to identify these sites, these really high value sites as people come across them.
Yeah, I think I'll just put a shout out to Jordan. I mean, he's worked hard on this project. He's produced a massive effort. We have a large number of contributors now. We just hit the 10,000 observation mark, so we're at 10,500 observations, and we're hoping that this becomes a real resource for people doing bee research. So if you want to find an aggregation of your favorite bee, we might know where it is if you visit the iNaturalist site. It's also a tool for conservation, and one of our goals is to kind of shift the focus away from planting a pollinator garden as the only solution to bee conservation to nest sites as a priority. So I think that it's kind of an underappreciated need that we should be focusing on nest sites and highlighting them and learning about them and protecting them.
It does raise this issue of managing these nesting beds. Here in the Pacific Northwest, alkali bees are, still a significant pollinator of, alfalfa seed. And in fact, it's hard to find nesting beds outside of these managed sites. It's an odd, way in which they have come under cultivation in some ways. And I have a slide in my presentation to growers saying the only managed ground nesting bee system in the world. I might have to revise that at this point. But those nesting beds are a lot of work to maintain. And this seems perhaps, the case that you brought to bear is actually little work. I'm sure the people who've been managing the cemetery have been doing so without the intention of managing these bees.
Yeah.
So, and I've also thought about this. I've had people tell me it's impossible to manage a long-term ground nesting bee bed because it will collapse because of the parasites. And this isn't always the case. So what can I have you speculate? What are the conditions that made this possible, and how might growers harness this information to, get better crop pollination outcomes?
Yeah, we're working on that. I mean, other folks are working on that to see whether it's possible to develop a kind of protocol for a bee bed. Imagine we could go to a grower and say, okay, take your backhoe and dig a 2 foot deep hole and fill it with sandy loam.
Mm-hmm.
And that would magically bring in a population of ground nesting bees. We're experimenting with that. I wouldn't say we've made a ton of progress. We do have bees nesting in our bee beds. It's not like we've created a massive aggregation, though. There's something mysterious about these aggregations. They clearly occur in areas with the right kind of soil texture, but there can also be sites with the right kind of soil texture and no bees. So there may be some chemical signaling that goes on. I mean, Jim Kane speculated on that years ago. Maybe there's an aggregation odor that brings these bees together. So I'll just leave it at that. We don't really know how to make a bee bed, but I think it's worth exploring, and it's certainly worth, working with growers in apple and blueberry and other crops like that to see if you couldn't identify one or two bee species, characterize their soil texture, and then build out a bee bed that would accommodate them. And maybe even transplant them. You can move buckets of soil around. We've tried that. You bury a bucket in an aggregation, come back a year later, dig up the bucket, move the bucket to another site. Now the bees that are in the ground come out. So there's all kinds of ways one could do this, but we don't have any definitive results yet, I'll be honest.
Well, it's the start of, something, and I see so much potential, and it really, even if it isn't directly applicable, it really opens our eyes to the natural history of these ground nesting bees that nest in aggregations. Thanks so much for reaching out to us about this paper, and I hope we'll have to have you on the show again soon to hear about some more wonderful natural history work that's coming out of your lab.
Thanks a lot. Thanks a lot, Anthony.
Thank you so much for listening. Show notes with links from each episode are available at the website pollinationpodcast.oregonstate.edu. I also love hearing from you, and there's a form at the website where you can pop in and say hello and give me feedback. If you want to support the show, remember to leave a rating on iTunes, Spotify, or whatever podcast mothership you use. And finally, if you have the means and you want to help support my lab's effort to document bee biodiversity in Oregon, visit OregonBeeAtlas.org and follow down to the donate button where you can make a tax-deductible donation to the Jerry and Judith Paul Native Pollinator Endowment. Every little bit helps. See you next episode.